It is my great pleasure to introduce Dr. David Teachey, MD, as our speaker for today. Dr. Teachey is Professor of Pediatrics and the Garrett Bordure Endowed Chair in Oncology at the Children's Hospital of Philadelphia and the Perelman School of Medicine at the University of Pennsylvania. He's an attending physician and an R01-funded physician scientist in the Division of Oncology at the Children's Hospital of Philadelphia, with clinical and research expertise focused on improving the understanding of disease biology and the translation of novel therapies for children with acute lymphocytic leukemia and rare inherited autoimmune diseases. He is a PI on multiple local, national, and international clinical trials and often performs the correlative studies for those trials in his lab. He is the chair of the ALL Disease Committee for the Children's Oncology Group and co-leader of the Immune Dysregulation Frontier program at CHOP. His CV includes over 200 peer-reviewed publications, including publications in Nature and the New England Journal of Medicine, and he has continuously been NIH-funded since training. He also has a history of training the next generation of clinician scientists, mentoring many students, fellows, and junior faculty who have embarked on successful academic careers, and his research and clinical expertise has been recognized by election into ASCI and APS. Dr. Teachey has worked closely with ALSF for years, first getting to know the Scots when Alex was a patient at CHOP, and Dr. Teachey was a fellow in training in the clinic of Dr. John Maris. Dr. Teachey has received multiple grants through ALSF over the years, including funding on the genomics of TALL that was recently published in Nature and Nature Cancer. So on behalf of ALSF, I wanted to thank Dr. Teachey for his time today, and we'll pass it over to him for his presentation titled New Insights into T-Cell Acute Lymphostatic Leukemia Disease Biology. And as a quick programming note, we'll have a Q&A session after the presentation, and you may ask questions to Dr. Teachey using the chat feature. So with that, Dr. Teachey, please take it away, and thank you for your time today.
Thank you so much, and thank you for the kind introduction. It's always, I always love seeing people from Alex's Lemonade Stand Foundation because you've done such great work over the years, and I really appreciate the funding for my own research and the research at CHOP in general, going back many decades now, I guess. I'm going to talk today about new insights into T-cell acute lymphoblastic leukemia disease biology. These are my disclosures. I won't go through them. Acute lymphoblastic leukemia is the most common childhood cancer, and it's common enough that relapsed ALL is the fifth most common cancer. It is broken down into two main types: B-cell acute lymphoblastic leukemia, which represents the majority of it, and then TALL, which will be the focus of today. I think the improvement in cure rates for children with ALL and TALL in particular is one of the great success stories of modern medicine. This is a disease that was uniformly fatal, even as 50 years ago, and nowadays the vast majority of kids are cured. Historically, children with T-cell acute lymphoblastic leukemia did worse than those of B-ALL, but with modern, more intensive therapy, patients with TLL actually have similar outcomes to those of B-ALL, although we're still not curing everyone, which is the goal. How did we kind of get here? How did we get to the point where we can now cure about 90% of children with TLL? It's really through using cytotoxic chemotherapeutic agents that were developed and approved between the 1950s and 1980. And we spent the past 40, 50 years just giving those same drugs in different combinations and different regimens and perfecting their use over time. And that's how we got to be where we are. And while we've improved outcomes in newly diagnosed patients using cytotoxic chemotherapy, we still do a very dismal job when patients relapse. So patients who have T-cell acute lymphoblastic leukemia that comes back, instead of having a 90% chance of cure, their chance of cure is less than a third. And we haven't really changed this number in a long time, and we need to do better. So we think of TALL as a disease that you get one good shot at cure, and we need to identify our best medicines and our best treatments and move it forward. So what I'm going to kind of talk about today is how we're thinking about and where things are moving forward on how we cure more children of TLL. Most of it, I'm going to spend time talking about disease biology. If I have time at the end, I'll talk about some new targeted therapies and immunotherapies being studied in TLL. We'll see how time goes. But I'm going to start by talking about improving risk stratification. And in risk stratification, we basically mean taking a combination of clinical features, disease biology, and response to therapy, and predicting how well patients are going to do based on data you obtain in the first month of therapy and applying it to put patients in different risk groups that we can tailor our therapy accordingly. And in B-ALL, we've been excellent at doing this, and it really has helped improve cure rates over the years. In TLL, in contrast, we're really not very good at risk stratifying, and currently the only feature that's used in risk stratification is response to therapy. But to try to do better, I'm leading a very large correlative project with a number of colleagues, and I'll go through some of them as we get to it. A big one being Charles Mulligan and his group at St. Jude Children's Research Hospital, where we did genomic profiling of a large cohort of patients with T-cell acute lymphoblastic leukemia, and what I won't talk about today, T-cell lymphoblastic lymphoma. That include bulk genomic sequencing, that included whole genome sequencing, whole exome sequencing, and RNA-seq at diagnosis, and whole genome sequencing of tumor and a whole genome sequencing of germline from 1,300 patients treated on the ALL-0434 clinical trial, and then another 500 patients treated on the 1231 clinical trial, the successor trial to that. And then also I'll show some data on some single cell profiling we've done as well on a large cohort of patients with TLL. We also doing a lot of work with proteomics, surfaceomics, methylation profiling, ex vivo profiling, and a lot of PDX work, and I'll talk about a smidge of that today as well. But the main focus will be talking about some of the bulk genomic and single cell genomic work that we've done. And then one question that comes up is, why are we even doing this genomic profiling? It's 2025. Hasn't somebody done this before now? And I think we've learned a lot of lessons as we did some of this large profiling of TALL. And one of the reasons we embarked on it was there's been very few studies in TLL that actually looked at a whole genome sequencing level or a single cell level in TALL. Most of it was using next-gen sequencing panels or whole exome sequencing. Another big problem with a lot of the literature that was out there before some of the work that we did was a lot of patients with relapsed and refractory disease. The high-risk patients are excluded, typically, or source of germline DNA for a lot of leukemia studies, including those from COG, as we use your remission marrow to actually isolate the germline DNA. Patients of TLL are more likely to have refractory disease. If you never get into remission, then you don't get that source of germline DNA with their remission bone marrow. So those patients were excluded from a lot of the trials and a lot of the studies on genomics that were published in the past. So the outcomes on those patients in those cohorts was significantly better than it was actually expected to be. And that's made all the published studies before this work underpowered to correlate genomics with outcome. And one of the things that I'll talk about a little bit today is some of the importance of non-coding alterations in the genome and why they're important in TALL. And it's just a brief overview. Most people think about with DNA and they think about genetics about coding mutations in a gene itself. And most of what we use in the clinic is actually genetic mutations in those coding parts of the genome. That's actually a very small percentage of our DNA, and actually most of our DNA is considered that junk non-coding alterations, which we've learned over time are far more important in disease biology, and in TAL in particular, they're highly important. And this is parts of our genome that actually regulate the coding parts of our genome. This is why our hair cells and our skin cells and our blood cells do not all look the same, even though the coding mutations are the coding parts of the DNA are the same in all those cells. It's the non-coding parts of our DNA that actually turn off and turn on different genes in different cell types. So through this work of sequencing over 1,300 children of TLL on a uniform clinical trial, we were able to make a number of observations, and we were able to group patients of TLL into 15 different genomic subtypes, which are depicted on the left, and that's a UMAP. Each dot is a case that's transcriptome sequencing. The closer they are in 2 dimensions, the more related they are. And for doing this, we were able to split TLL into 15 subtypes with distinct genomic drivers, distinct gene expression patterns, developmental states, and different outcomes. Some of these were novel, and through this we proposed new nomenclature. And again, this was a project that I led with Charles Mulligan, and a lot of the work was done by a very talented postdoc, Petri Polonen. We were able to identify the primary leukemic driver in the majority of samples tested. Over 95% of patients that we tested, we were able to identify the driver. Most of these were things that we knew about in TALL, things like TAL1 or TLXL3 or LMO2. What we know better now is the frequency of some of these. There are a small percentage of cases we weren't able to identify the leukemic driver. We're now in the process of doing long-read sequencing to see if we can identify that in a subset of those patients. We were also able to get an overall scope and spectrum of the genomic landscape of TLL. This is looking at different genetic alterations in TLL. Most of the genes that we found that were altered in TLL are ones that we expected, like Notch1 and CDKN2A. The difference is that we actually understand the frequency better and the type of alterations. We did identify 13 novel genes that were recurrently mutated in TLL that hadn't been previously described, but most of the genes that we found that were altered in TLL were ones that were previously described. But one of the major observations we made from this work that wasn't known beforehand is how common non-coding alterations are in TALL. So what we found was over 50%, almost 60% of leukemic drivers, the thing that led to leukemogenesis, were genetic alterations in non-coding or intergenic parts of the genome. So if you just did whole exome sequencing or you did next-gen sequencing approaches, you would miss the primary driver of leukemia, the main thing that was making it happen, the thing that we'd use for outcome or risk stratification by using those kind of sequencing approaches. And it's one of the main reasons why we've learned not that much on TLL disease biology over time, and more importantly, why we haven't been able to risk stratify patients of TLL because we've not been looking in the right place. It is very complex. As you can see here, this is looking at all the different types of non-coding alterations and all the mechanisms of oncogene activation that we saw, and the number of different genes that could be involved. So just something like TCR enhancer hijacking, which means that you have the enhancer of one gene that's activating another one, and this is in the T cell receptor, could be done by over 40 different oncogenes and in different number of cases. But so it's not like B-ALL or a lot of other cancers where you can put patients into a nice small number of buckets. The biology is far more complex in TALL, and we have a lot of work to do to understand it better. We did verify some of these non-coding alterations mechanistically. In some of these cases, we used HiChip, which is very good at identifying enhancer hijacking and super enhancers and basically how an enhancer activates a promoter region of a different gene. This is just showing that I won't walk through it just for time. 3 of the cases that we used HiChip to actually confirm some of those non-coding alterations were actually real and biologically meaningful. Other types of non-coding events that we actually saw in addition to enhancer hijacking and super enhancing, this is just 2 examples here. One was looking in Notch, and I'll get to this later, why it's important. We found that in a percentage of cases, we found intronic SNVs that actually led to increased coding sequence of one of the exons that actually increased Notch activity. Another example of some of the non-coding biology that we saw here was in the IL-7 receptor. We found that a deletion of the transcriptional start site and led to a loss of the mutated allele, and then you end up having enhancer hijacking by the AL7 receptor to PLLR, and that led to increased JAK-STAT signaling. So we're starting to understand the biology of some of these non-coding alterations a little bit better. The other thing that we were able to do was we took our different genomic expression profiling and we compared that to a single cell map. And for this, we correlated with the Kai Tan lab, and this was work that was funded by Alex's Lemonade Stand, where we did single cell genomic profiling on healthy normal bone marrow and healthy thymus from different children to actually get a normal single cell map of normal cellular repertoire, which is shown on the left, where instead of where each dot is a different cell for those different bone marrows and thymuses that we sequenced. We then took our genomic data and we overlaid that to see where was the potential cell of origin for different subtypes of TALL. You can't actually prove it's that, but at least what lineage were they most similar to, which would infer they may have arose from those earlier lineage states. And to show some examples of that, and this is actually how we proposed some of the new nomenclature for TALL, was based on this lineage or potential cell of origin. So for example, we renamed one group of TALL, which was formerly known as Tau1RA, and we renamed it Tau1 double positive like because at a single cell level, these cases actually mapped with cycling double positive T cells. In contrast, there's this other group which has Tau1 dysregulation that we term Tau1 alpha beta like. And the reason we did this is because those cases mapped more with alpha beta T cells. So it was actually the lineage that actually drove, and that was actually seen in the different clusters that they had. Another example of that was a very rare group that we identified, which we named the ELMA2 gamma delta like, because in these cases, they actually mapped with gamma delta T cells. So in addition to naming some of the groups based on where they mapped in a single cell map, some we basically renamed or named based on the primary oncogenic driver, because that was the main driver in almost every single case. Some of those are shown here. I won't walk through them, but there are things that we knew before, like BCL11B, TLX1, TLXL3. And then we were able to, with having as many cases as we have, actually dive down deeper into some of the biology there and actually see what was activating, for example, in the TXL3 group and what was causing the dysregulation. And in TXL3 group, we found it was most commonly like BCL11B enhancer hijacking, but we also identified a number of arrangements with TCR beta, CDK6, and other partners as well. One thing that we really wanted to do in addition was to understand more and to understand the biology of early T cell precursor ALL. So currently, the only thing that's used in the clinic to distinguish different subtypes of TALL is its immunophenotype, so antigens on the surface of the T cells. And about 20 years ago now, Dario Campana, when he's at St. Jude's, described a subtype of TLL, which he termed early T cell precursor ALL, which had a unique immunophenotype. And in initial studies, they looked like they had really inferior outcomes. With more modern therapy, we found that the outcomes for patients with ETP immunophenotype are actually similar to non-ETP immunophenotype, but there's been some discrepancy between different cooperative groups where some groups, like the Children's Oncology Group in North America, we found that ETP ALL immunophenotype is not affiliated or associated with inferior outcome versus in adults it had been. And we wanted to understand more of why we're seeing that discrepancy. So we basically looked at the genomics of patients with an ETP immunophenotype, and we found that those cases clustered in one of three groups. They clustered in the TXL3 group, they clustered in the BCL11B group, or they clustered in this group that we renamed as the ETP-like group, and I'll explain why we named it that way. There's another immunophenotype that was described in ETP-AL that was similar to ETP-ALL, first described by Brent Wood, which basically has a similar immunophenotype but is missing one of the criteria of being CD5 negative or dim. And for this subset of cases, we found they also clustered in the TXL3 group, they clustered in the tau-1 alpha beta-like group, and they also, most of the cases, clustered in this group that we named ETP-like. If you looked at the entire cohort of 1,309 cases, we found that 70% of the ETP immunophenotype and 40% of the near ETP immunophenotype clustered in this, what we called ETP-like subgroup that they clustered together. If you looked at it a different way, within the ETP-like subtype, a third of them had ETP immunophenotype, a third of them had near ETP immunophenotype, and a third of them had non-ETP immunophenotype. So it was actually a balance of the three different immunophenotypes, even though if you looked overall at patients of ETP immunophenotype, a lot of them were in this group. We found if you looked at outcomes, and this is looking at a Kaplan-Meier curve of those 15 different subgroups that I mentioned, and I'll get to some of the outcome data in a few slides, we found that patients with ETP-like ALL, this very large group had inferior outcomes compared to many of the other groups with an event-free survival of around 75% compared to the 85+% we see in most patients. And then if you look within this ETP-like group, we found similar genomics regardless of immunophenotype. We saw similar pathways that were dysregulated. We saw similar poor MRD response, similar rates of induction failure. So basically, in ETP immunophenotype, there's been a lot of studies that have shown certain pathways are dysregulated in some cases. Some of these patients are more likely to have refractory disease. What we found is it's actually these patients in the ETP-like group that actually have these similar biologic features regardless of immunophenotype. And if you looked in the ETP-like group, their outcomes were the same regardless of ETP immunophenotypic status. Kind of a better way to kind of take it home is if you look at the genomics and break down patients based on their immunophenotype into the ETP-like group versus all others. It's shown here. So on the far left is a Kaplan-Meier curve looking at patients with ETP immunophenotype, dividing those with ETP-like genomics versus any other genomics. In the middle is near ETP immunophenotype, same kind of split. And on the far side is not ETP immunophenotype, same kind of split. And what you can see is the patients who do poorly are actually not the ones defined by their immunophenotype. They're the ones that are defined by their genomics. So we've kind of drilled down and identified who are the high-risk players in patients with TLL, and a lot of them fall into this ETP-like subtype group. Another way to look at it is kind of a Venn diagram is if you look at it by immunophenotype, you can define patients as ETP, near ETP, or non-ETP immunophenotype, but they're genomically heterogeneous, and it doesn't really correlate with outcome, versus if you do this by its genomics in the ETP-like group, which is made up of a combination of the different immunophenotypes, it's much better at predicting biology and, which we really care about, disease outcome and chance of being cured. We can take this relatively large subgroup of a little over 200 cases out of the 1,300 and break it down into different driver lesions and actually different subgroups, which are kind of shown here, where we further the ETP-like subtype is frequently made up of clusters of MLLT10, ETP6 lesions, KMT2A rearrangements, NUP98, and other alterations as well. Another group that we actually identified in the last group that I'll mention as far as this genomic profiling of the 15 different groups, we named TME enriched or tumor microenvironment enriched. This is a group that we actually found no unifying driver, despite the fact that they all clustered together based on RNA-seq. This group did have high levels of tumor microenvironment and was heavily enriched with monocytes. Importantly, they had low blast at diagnosis. So it could be that the reason weren't able to identify a driver is just the blast percentage was relatively low. But still, in most of these cases, it was like 30 to 50%, which should be enough to identify a leukemic driver. But we are doing long read sequencing on this group to see if we can identify the leukemic driver to understand it better, to see if it really is a unique subtype or they are just patients with low blasts. And if needed, we'll actually get more additional samples from this group and sort them to actually look at the genomics. One of the things that was very exciting, I'm very happy to see that came out of this collaborative work was we're finally able to really integrate genomics with outcome and predict who's going to do poorly in patients and who's going to do well in patients of TLL, so we can start thinking about tailoring therapy. For a lot of this, we collaborated with Stan Pounds, who's done a lot of work on genomic outcome and profiling and risk model development. A couple things that we found here, there were some groups like this LMO2 gamma delta, like I mentioned before, and a SPY1 group who did very poorly with really dismal outcomes. Fortunately, these two groups were rare, with only 10 and 11 cases respectively in each of the two groups. We did find, even though these two groups had poor outcome, that didn't necessarily correlate with MRD response, where the LMO2 gamma delta-like group was basically had a lot of patients had refractory disease. The SPY1 group, actually, most patients actually responded to chemotherapy very favorably, and they just had a lot of relapses. The ETP-like group that I mentioned before has inferior outcome overall, but when we started drilling down on some of those different subtypes based on the driver or different clusters like KMT2A and MLL-T10, we did find some differences that some of these groups did really bad, but some of them, like the ZLFP36L2 group, actually had pretty good outcome. Despite differences in EFS, which is shown on the left, they all had poor MRD response, which is another thing that shows that MRD is not necessarily a great association with EFS or predictor of your overall survival or event-free survival. Another thing that we noticed is there was a strong interaction between your expression group and your genomic clustering and the driver lesions. So for example, we found that patients with MLL-T10 alterations who clustered outside of the ETP-like group actually had very good outcome with 5-year survival of around 93%. In contrast, patients with the same driver alterations, MLL-T10 alterations, who were clustered within the ETP-like group, those patients did very poorly with 5-year survival around 73%. We saw similar with other comparisons if you looked at patients who had different drivers with and without this ETP-like group. For example, KMT2A rearrangements, those who were in the ETP-like cluster did poorly. Those outside the ETP cluster, the KMT2 rearranged patients who were not ETP-like actually had 100% survival in our study. And which I will not walk through, we were able to identify a number of different genomic alterations, not just looking at the 15 subgroups that were predictive of event-free survival, overall survival, and disease-free survival. And there was a lot of overlap there. Another thing that we identified was enrichment of this SPY1 group, that rare group that I said before, in patients who had were classified as secondary malignancies. And a large percentage of these secondary malignancies that were seen on 0434 clinical trial were in this group. But if you actually started looking at them, a lot of them were different types of myeloid sarcoma and histiocytic lesions, not the kind of things we traditionally think of as secondary malignancies. If you looked at those at a single cell level, the SPY1 group, they actually mapped more thymic dendritic cells. And that raised the question is maybe the SPY1 group isn't really that they're having a secondary malignancy. Maybe this group, when they relapse, it's actually more of a lineage shift. And we looked at that at a molecular level. We did some collaboration of groups to get some more relapse samples from some of these cases. And what we showed was these patients, as they have the disease come back, they acquire some genetic alterations that are more similar to histiocytic lesions. And basically, these patients aren't developing a secondary histiocytosis or myeloid sarcoma. Their TALL is changing to what looks like histologically as a histiocytic lesion, but is still biologically the TLL. And these patients need to be treated as if they have TLL that relapsed and not as if they have a second or a new cancer. Making it even more complicated, though, as we start to think about the biology and outcomes in TLL, we found that it wasn't just the gene that was altered actually correlated with outcome, it was how that gene was altered. So as an example, Notch1 alterations are one of the most commonly altered gene in TLL and are usually considered a favorable prognostic lesion. And what we found was that most patients who had a Notch1 alteration that actually inferred favorable outcome. This is looking at cumulative incidence of relapse. So opposite of the way the curves look in a Kaplan-Meier curve. However, we did identify this group of patients who had Notch1 intergenic loss, so they had Notch1 alterations that were non-coding, and those patients actually had worse outcome. And Notch was not the only gene that we saw this in. We saw this actually in quite a number of genes where it wasn't just if the gene was altered, it was how it was altered that actually impacted its outcome. Things like PTEN, MYC, and others actually had a strong association of outcome based on the type of genomic alteration. All that said, we were able to integrate our genomic data and the 15 different genomic groups and different genomic alterations with clinical data, including white cell count at presentation, CNS status, and disease response to make some risk stratification multivariate models. This is just showing one example of that, where we can actually group patients with TLL into 4 different buckets with EFS ranging from 65 to 97%. The problem here is you need whole genome sequencing and RNA-seq to do this risk stratification, which is actually not the most easy thing to do in the clinic currently in 2025. This was another risk stratification model that we developed, which basically mainly focused on some clinical variables and RNA-seq in those 15 different subgroups and some of the subtypes that I mentioned before. And through this, we're able to risk stratify patients into 8 different groups with EFS ranging from 98% to 45%. Those patients with 98% survival, we are overtreating, and maybe on future trials, we can ask some de-intensification questions. And the patients with survival in the 45% need alternative therapies like CAR T cells or a transplant, because we're obviously not doing a very good job with chemotherapy alone. And we're not the only group that's actually been doing some exciting work in TLL genomics lately that's actually showing a correlation with disease biology and outcome. There's been a bunch of papers in 2024 and 2025 based on some of our colleagues in Europe using next-gen sequencing, methylation, and other types of genomic profiling to correlate that with outcomes. And other groups as well that I'm not going to go through all these data, just to say that we're not the only one who's studying T-cell genomics out there. There's a lot of other groups doing a lot of exciting work. Beyond bulk sequencing, the next thing that we really wanted to do was start studying TLL at a single cell level. And this work was all supported by an Alex's Lemonade Stand single cell grant. And for this, we did single cell profiling on a large cohort of patients of TLL and compared that to some other types of leukemia as well. For this, one of the things that we did is we actually took our different TLL samples and we did single cell RNA-seq and we did single cell ATAC-seq. And we then took the cases and we mapped them against our normal lymphocytes using that single cell map that I mentioned before, using healthy thymus and also using healthy bone marrow. And we compared patients as a start that had ETP immunophenotype with near ETP immunophenotype and non-ETP immunophenotype. And we were not surprised to see that patients of ETP immunophenotype actually had a more earlier lineage arrest as far as where they actually associated on that lineage map. We found that ETP ALL was actually more close when we actually compared it to the healthy reference to T myeloid mixed phenotype leukemia and not similar to AML, which actually was more on the myeloid lineage as we'd expect. One thing we really wanted to use these data, though, for was to understand the biology of refractory disease in high-risk populations and relapse. So for this, we did single-cell profiling at diagnosis, but we knew their eventual outcomes. So we enriched for patients who had poor response or they had relapse disease. And this is showing an example comparing patients with high MRD at the end of induction, so they were induction failures, versus patients who had a good response and looking at their diagnostic blasts. And what we found is patients who had eventual refractory disease had enrichment of what we called a bone marrow precursor-like population. So they were actually more early lineage as far as compared on an early lineage map, showing some differences biologically between those who would later have a good versus poor response to chemotherapy. When we compare differentially expressed genes from patients who had this BMP-like signature versus ones who did not, which were more like mature T-cell signature, which we call T-specified blasts, we saw some differences in patterns of differently expressed genes. The ones that were more T-specified, we saw the things we expect to see in TLL, notch targets, things involved in core T lineage commitment. On the contrast, the ones that were in this BMP-like group had things that were involved in like self-renewal and recovery from genotoxic stress. We then took the differentially expressed genes in this BNP-like refractory population, and we integrated those into different drug targeted databases to predict which drugs these may be sensitive to. And for that, we predicted a number of different targeted therapies that may be effective against these BNP-like blasts. And one thing that I should have mentioned that I forgot to mention earlier on is this work was done in collaboration with Kai Tan's lab here, and a lot of it was done by a very talented MD PhD student, Jason Zhu, who just started residency this week at Stanford. Unfortunately, not in pediatrics and internal medicine, but he'll be a superstar and do great. To test some of those drugs that we actually predicted may be good for patients with TLL with this BNP-like signature, we made PDX models from a large number of cases that we did single-cell profiling on. The first thing we wanted to do was to make sure the BNP signature actually was maintained in the mouse models and the PDXs after they engrafted, which we found that it was. This is just showing an example in a handful of cases. We then took blasts after we passed them through the mice, and we sent them to Jun Yang at St. Jude, who does drug profiling and allows us to take like 50 to 60 million cells and test 50 different drugs in a petri dish to see how well they work. And what we found was for the BMP-like TALL, they were resistant to chemotherapy, is kind of what we saw in the clinic, but they were more sensitive to some of these drugs that we predicted they would be sensitive to, like venetoclax and navitoclax. If you compare those to the cases that did not have this BNP-like signature, we validated a little bit of this using the PDX models, actually treating the mice themselves, showing a nice correlation as well. So starting to see some correlation of why we're seeing poor outcome in these patients and what novel therapies may be used to treat these patients instead. We found that one of the reasons these patients probably do worse with therapy is that they had a change in expression of the steroid receptor, which has been shown to correlate with steroid resistance in cases of TLL, and those with the BNP-like BAS were predicted to be steroid resistant, which they were, ex vivo at least. And then we took this signature that we only saw at a single cell level. We did not see this when we did our bulk genomic profiling. And we were able to, and Jason was able to drill down and actually identify 17 genes that were able to predict the signature. We then took this 17 gene signature and we applied it to our bulk genomics from the 0434 cohort that I mentioned before, and also successor trials, some of the early genomics we've done on that 1231. And we found that these 17 gene signatures able to accurately risk classify patients on both of the trials and of different immunophenotypes. So a signature that we identified for the single cell omics that we didn't see in our bulk omics, we could then apply to the bulk omics once we knew what we were looking for. That actually may be a better thing that, or an easier way of predicting outcome in patients with TLL, because this is something that could be easily done in the clinic tomorrow. One question that does come up sometimes when I show these data are now we've described two new subtypes. There are two new types of biology in TLL. One is this ETP-like thing, and another is this BMP-like thing. And are they the same thing, and how much do they overlap? There's been a couple of other papers that have come out by other groups recently, either using our data or their own data, that have also found these early refractory disease signatures, and we've kind of compared those as well. So one thing we did was actually compared outcomes in patients with ETP-like and BNP-like based on the status. So basically taking patients with ETP-like who are also BNP-like versus ETP-like that were not BNP-like versus BNP-like that were not ETP-like. And this is showing survival curves there, showing that the patients who do really poorly are actually the ones who have both. They fall in the ETP-like genomic group and they have the BNP-like signature. We did find the BNP-like signature is enriched, not unexpectedly, in the ETP-like group and also in the BC11B group, but was also found in other subtypes of TLL as well. And we're not the only group that's done single-cell profiling of TLL recently. Similar to some of the bulk genomic stuff that I showed earlier, there's been a number of other exciting publications that have recently come out from another groups doing single-cell profiling, showing a lot of the same biology, which is always nice to see people replicate some of your work. And some other stuff that's in press are actually out in the literature now as well. So I think it's an exciting time with modern genomic technologies in TLL because we're really learning a lot of insights into TLL biology that I think will directly impact patient care in the clinic, both through better risk stratification and also target identification for novel therapies. And then, as I mentioned at the very beginning, we also are doing, to understand some of this biology a little bit better, we have ongoing projects where we did methylation profiling on a large cohort of cases. This is on the left is showing a methylation map that Petri's been working on. Some of our preliminary analysis says we actually may be able to predict our 15 different genomic subgroups by their methylation signature, which is very easy to translate in the clinic because methylation signatures are already used in the clinic in brain tumors and some other cancer surface stratification. We're also doing mass spec proteomics to actually understand the impact of some of these genetic non-coding alterations, both at the transcript level we've already been able to look at, but actually to look to see how it impacts protein as well. And then finally, we're actually sequencing a large cohort of cases with T-cell lymphoblastic lymphoma. We used to think TLL and T-cell lymphoma were the same disease, and we've now learned in the clinic, as we've been starting to use new therapies, that they're different, and we are seeing some differences in genomic profiling in TLL versus T-cell lymphoblastic lymphoma. And this is some work that's been done in the lab by Lahari, Rowan Shame, and Haley Newman. I'm going to kind of shift gears now for the last few minutes and talk about a couple other topics on ways that we're trying to improve cure rates in TLL. One is to improve understanding of healthcare inequities and inferior outcomes in historically disadvantaged populations. There's been a lot of studies that have looked at the impact of self-identified race and ethnicity on outcomes in patients with acute lymphoblastic leukemia as well as other cancers. Surprisingly, some nice data published by Sumit Gupta, which reviewed our experience in COG over time, found that patients who self-identify as non-Hispanic Black or as Hispanic have worse outcomes. Surprisingly, though, we did not see the same thing in TLL. In TLL, we didn't see any differences outcome based on self-reported race and ethnicity. Self-reported race and ethnicity are really social constructs, and we wanted to look at this as a biologic construct. And for this, a very talented junior faculty member in the lab, Haley Newman, did a lot of this work. It was actually it was her idea to go here and actually do some of this study. And looking at genetic ancestry is actually taking SNPs in your germline DNA and looking to see where your germline DNA originated. Is it more like somebody from Europe? Is it more like somebody from Africa or different parts of the world? And you can use it as a genomic level, so you're not looking at this more of a social determinant of health. You're looking at their disease biology to see if there's an interaction between that and outcomes in tumor genomics. One thing that stood out right away was we found that patients of Native American descent who were with BALL had worse outcomes. But we found the opposite in TLL, where those patients actually had superior survival. We compared the frequency of those different genomic subtypes that I mentioned before based on ancestry. Not surprisingly, we we're kind of expecting to see this. We saw differences in frequencies based on your germline ancestry. For example, if you're of African descent, you're far more likely to have the ETP-like signature than if you're not of African descent. But we were not expecting to see, and I think the most important thing that came out of the work is we found that the prognostic impact of different genes varied by genetic ancestry, and this is just showing one of them. Notch, I mentioned before, typically associated with favorable outcomes. If you look on the far left, it's a Kaplan-Meier curve looking at genomic alterations in Notch pathway if you're of European descent, and they actually have favorable outcome. If you're of American Indian descent, also a favorable outcome. But as you can see on the far right, if you're of African descent, having the similar Notch mutations do not infer favorable benefit at all. So a Notch pathway alteration in somebody of African descent has no impact on prognosis in either direction. But if you use Notch to risk stratify patients, you would misstratify or misclassify patients of African descent. And we found in TLL that Notch was not the only gene, and we found that over 80% of patients with TLL have a genomic alteration in a gene where the prognostic impact of that gene varies tremendously based on your genetic ancestry. So a very strong association between germline ancestry, somatic tumor genomics, and outcome that we really need to understand the disease biology of that better. And why is that important in the clinic? It shows that we have the potential to misclassify patients. So most of our risk classifiers that we use in the clinic are based on studies that happen in North America or Europe, and most of the patients there are of European descent or Caucasian or white. And those actually drive the risk stratification models. This is a model that was developed in Europe using next-gen sequencing to risk stratify patients with TALL. And we took our 0434 cohort and looked at this classifier based on ancestry. And on the left, if you're of European descent, their classifier is actually extremely accurate. In the middle, if you were admixed American, American Indian descent, we found it was also very accurate. However, if you were of African descent, this risk classifier actually misclassified you, and certain patients that said were favorable actually had worse outcomes, and vice versa, primarily because Notch was actually used as a big gene in this prognostic model and risk classifier. However, we were, though, when we tried to take a step back and say if we wanted to make ancestry agnostic risk classifiers that would accurately risk classify patients regardless of genetic ancestry, we were able to do that. And this is just showing one example of that, where a risk classifier that accurately classifies patients regardless of your genetic ancestry. And I think this is a lesson for just cancer genomics as a whole, that we need to make sure when we study biology and we integrate that with outcomes, that we consider ancestry in our models so we don't misclassify patients that are historically disadvantaged, and actually increase disparity in those populations. And then the last thing I'm going to talk very briefly before I open up to questions are some of the work that's going on, new targeted therapies and immunotherapies in TALL. There are a lot of potential targeted therapies for TLL that are very exciting right now. I think one of the tough parts of targeted therapies for TLL is for most of these, there's only a small percentage of patients who actually have dysregulated signaling in JAK-STAT or other things, so they won't apply broadly. Two clinical trials that tested novel therapies in TLL in the front line were published in the past 20 years. One was the 0434 trial, which I mentioned before. This study had a 2x2 pseudofactorial randomization comparing nelarabine versus no nelarabine and two different types of methotrexate, where we found that nelarabine improved disease-free survival in patients of TALL but not T-cell lymphoma. The successor study to that was the 1231 study that I was the study chair on. This study randomized patients to receive or not receive the proteasome inhibitor bortezomib during induction and delayed intensification. What we were surprised to see, and was totally unexpected in this trial, is we found that bortezomib did not help patients with T-cell acute lymphoblastic leukemia, but was very effective and improved both event-free and overall survival in patients with T-cell lymphoblastic lymphoma. I'm going to show that just based on time. We had been treating patients with TALL and T-cell lymphoma the same way in the US and abroad. However, some of these data actually changed the way we treat it, when nelarabine is now standard of care in patients of TLL in the front line, and in patients of T-cell lymphoblastic lymphoma, bortezomib is now part of standard of care. And we're studying a lot of different molecular targets and targeted therapies in preclinical models, and there's some ongoing early phase clinical trials, one of which is targeting JAK-STAT inhibitor, targeting the JAK-STAT signaling pathway focused on ETP immunophenotype, or probably more appropriately ETP-like. This was some early work from my lab that was done by Shannon Maud and others, where we showed that patients with ETP frequently had dysregulated JAK-STAT signaling, and if you use JAK-STAT inhibitors in preclinical models, they were highly effective. And there's now some early phase clinical trials testing JAK-STAT inhibitors in ETP ALL. We can use some of the genomics that I mentioned before to actually look to see if there's different regulations and different pathways based on genomic subtypes to predict which pathways may be dysregulated at relapse and which novel therapies that you may consider. This is just showing the arrow is pointing at JAK-STAT alterations based on your genomic subtype, showing that in some groups like BC11B, you're highly likely to have a JAK-STAT signaling abnormality, versus others like KMT2A, you're very low likely to have a JAK-STAT signaling alteration, which could say that impact your decision to use a JAK-STAT inhibitor in the relapse setting if you're trying to get a patient a transplant. Another area of exciting targeted therapies in TALL is some work that first was done by Jean Picard-Boquin, followed by Jun Yang, where they found that about 40% of cases of TLL in a petri dish are sensitive to dasatinib, and it wasn't due to having Abel class fusions or alterations. What they found was these patients that were sensitive to dasatinib frequently had activated SRC signaling through activated phosphorylated LCK. It's about 40% of cases of TLL. And there now are some studies that are actually looking at integrating dasatinib, a tyrosine kinase inhibitor that hits LCK and SRC, in clinical trials, and we're very excited to see how these data look. There's also been some exciting work in the targeted therapy space, looking at targeting the apoptotic machinery, some preclinical studies showing that patients of early lineage TLL are more likely to be BCL2 dependent. Those of later lineage TLL are more likely dependent on BCLXL, and that correlates biologically and response ex vivo in the clinic to BCL2 inhibitors versus BCLXL inhibitors. Wendy Stock ran a trial that included some kids that included a BCL2 inhibitor, venetoclax, as long as a BCLXL inhibitor, navitoclax, and treated a few pediatric patients who were relapsed that showed some pretty nice response rates around 70%. And there are some current trials ongoing and some data that have recently been published looking at targeting the apoptotic machinery with chemotherapy or demethylating agents. Unfortunately, the venetoclax is no longer available because it didn't work in an adult cancer. One of the things that we see in peds onc all the time, which is some drug is being tested in adults, doesn't work, so then it goes away for kids as well. Another targeted therapy that's actually being explored in the clinic right now is targeting cyclin-dependent kinases. This is some work that Karen Bride did in my lab years ago, based on some stuff that came out of Ioannis Iofannis' lab, showing that cyclin-dependent kinases are frequently dysregulated in TLL, and that correlates with response to drugs like palbociclib and ribociclib, which are commonly used in females with breast cancer. And Elizabeth Rates recently ran a trial that added palbociclib to chemotherapy for patients with relapsed refractory disease, and very small n, but there were some patients of highly refractory and multiply relapsed TLL that actually went in remission with palbociclib plus chemotherapy. And the last area that we're really studying right now in the clinic is trying to get immunotherapies into the clinic with TLL. There's been a lot of work in this area trying to first figure out what should we target in patients with TLL. Probably three of the highest expressed antigens are CD5, CD7, and CD38, which has led to a lot of the studies testing different immunotherapies targeting those antigens. We've kind of looked to see if there's differences in expression of those antigens by genotype. We have found some differences. For example, CD7 versus genotype, you can see some of the differences. We'll see if that actually impacts response to CD7 CAR and other immunotherapies in the clinic. We looked at a naked monoclonal antibody, daratumumab, which is an anti-CD38 monoclonal antibody used in myeloma. We first studied that in preclinical models a few years back. Postdoc Karen Bride, who's now a faculty member at Harvard, showing that it was highly effective in preclinical models of TLL. This led us to open an international phase 2 trial, the FINISH trial, that was chaired by and run by Laura Hogan and Tina Botla, just published in Blood last year, where we added daratumumab into relapse therapy, and we found a very impressive over 80% remission rate after 2 blocks of therapy when we added daratumumab into our chemotherapy backbone. And that has led to our next clinical trial, a phase 3 trial that should hopefully open this summer or this fall at the latest. That's going to be chaired by Keith August and Eric Schaefer, vice chaired Marie-Louise Solis and Stephanie Lim, which is randomizing patients of T-cell leukemia and lymphoma to receive or not receive 15 doses of daratumumab added to an intensive cytotoxic chemotherapy backbone, the first real use of immunotherapy in the front line in patients of TLL. And I think the other exciting area in TLL and immunotherapy that's ongoing right now is CAR T cells. As people know, CAR T cells have really transformed the landscape in patients of B-ALL, have not been as effective in some other cancers, but are being studied in a lot of cancers currently and seeing some nice results. For example, the GD2 CAR that was studied by Franco Locatelli in Italy, some discouraging results looking at CAR T cells in AML, but the TLL data are looking very similar to what we're seeing in B-ALL. So this is an example of a study from China where they used donor-derived CD7 CAR T cells. So they basically took T cells from a donor, from a patient who had a transplant or is about to have a transplant, made CAR T cells with those and gave it to the patient. And they found that 19 to 20 patients went into remission. Some of those remissions have been durable. There's been work looking at allogeneic or off-the-shelf CAR T cells for TALL, one that was developed in collaboration with my lab and Beam Therapeutics, is a CD7 CAR that we knocked out using base editing 4 different genes, the T cell receptor, CD7, CD52, and PD1, highly effective in preclinical models. We opened an adult trial that treated 5 adults. 4 of them went into complete remission, and the pediatric study for this just opened. Carolyn DeOrio is the study chair for this. It's just a single institution right now study at CHOP. And the first patient was treated this month. The second patient is getting treated next week. And just showing proof of principle, this kind of technology works. There was a nice study that used a very similar base edited CAR at Great Ormond Street, where they treated a couple kids with TLL that were published in New England Journal of Medicine 2 years ago, showing it was highly effective in those small number of kids. And then some nice, the last CAR I'll mention is from Dario Campana's group, who published some nice work doing a more traditional autologous CAR, where he used a protein expression blocker to prevent CD7 from getting expressed on the surface of the CAR T cell. One of the issues with CAR T cells for TLL is fratricide, where basically if you're attacking something that's on T cells and T cell blasts, that's also on normal T cells, the CAR T cell may attack itself. More of an issue of some antigens than others, but this was an elegant way to overcome that with CD7. And if this CAR T cell, similar to the other ones I've shown, had a over 90% complete remission rate, and some of those remissions have been durable. So a really exciting time in TLL where we're actually seeing some immunotherapies make an impact in the clinic. And there are many other CAR T cells that are being in development and in different stages of early clinical trials in TLL. So in summary, there's a lot of exciting stuff going on right now to improve cure in TLL through understanding disease biology, trying to understand healthcare inequities and how to mitigate those better, and incorporating new targeted therapies and immunotherapies in the clinic. One person I should have mentioned that I didn't mention in some of the work on healthcare inequities, we collaborated with Kira Bona at Dana-Farber Cancer Institute, who's a health equity expert and has been helping with some of the work in that space. So with that, I'd like to thank the members of my lab that are shown here. Carolyn Durio did some of the work with some of the immunotherapy. Some of the genomic work was done by Ron Shame, Laura Hari and Haley, as I mentioned before. Jason Zhu, talented postdoc, now a resident at Stanford, who did a lot of the work with the single cell profiling. A lot of members in COG that we've worked with for a lot of clinical trials. We won't go through them all. And then I've had the fortune of having a lot of great research collaborators over the years to do a lot of this work, because it really takes a village to pull some of this stuff off. In particular, thank Charles, Kai, and June, as well as Petri. And with that, thank funders, including Alex's Lemonade Stand, and most importantly, patients and families. Thanks.
Thank you, Dr. Teachey. That was a fantastic presentation. A lot of great findings there. So now we'll open it up for Q&A. We have about a little over 5 minutes for Q&A. So if you have any questions, feel free to post them and I'll read them to Dr. Teachey. So our first question is, when you say that the type of mutation within a gene makes a difference in outcome, were they all loss of function mutations, and what types of mutations were more likely to lead to worse outcomes?
So it's a great question. One, it depended on the gene. And then what we were not expecting, and we still have to figure out, is they weren't, so for Notch, they weren't loss of function. So we found that the alterations that were inferior outcome were intragenic ones, so non-coding alterations, versus most of them were coding. And when we actually looked at the impact on Notch expression, we found it increased it. We actually have some nice data that shows that there's a strong correlation between Notch expression and outcome. So usually more Notch signaling equals good thing. For some reason, though, in this subset of patients who have non-coding alterations, they had even higher expression, and those patients actually did worse. So it could be how much the pathway is activated, where a little activation is good. If you activate it too much, that's bad, but we really need to understand the biology better. And we're trying to use some of the proteomic stuff. That's one of the big questions we're looking at the mass spec, is actually how is it impacting things at the protein level. Similar for other genes, it depended on the gene, as far as some of them, yes, it was more straightforward, like gain of function, loss of function, but some of them, the biology is far more complex and we're still trying to sort it out.
Great, thank you. So second question is involving your SPY1 classification. So you mentioned that what was originally thought to be a secondary lesion is actually a relapse. Is that specific to the SPY1 subset of TLL, or do you think there's more subsets that we don't yet know? And how widespread do you think this is in diagnoses across different cancer types, even independent of TALL?
Great question. We saw it primarily in the SPY1, but we also did see there were other groups that seen as well, and there are reports in the literature of patients of TLL relapsing with histiocytic lesions that were thought to be histiocytic, that you would now wonder if those were also relapse of different genomic subtypes. So I think it's beyond that. I think it's just enriched in that, and probably SPY1 is at a higher risk of that because it really probably comes from a dendritic cell origin, so it's already on that cusp of wanting to go down a histiocytic lineage versus going down a T cell lymphoblastic lineage. My guess is it probably does occur in other cancers as well, especially ones that have a lot of potential for lineage plasticity. We see that with immunotherapies. We're seeing lineage shift or lineage switch as a big thing that in the immunotherapy era, where cancer finds a way to get around it. get around things. And I imagine that histologic or lineage switch is probably just one mechanism that certain cancers use to get around the chemotherapy that we're using and relapse.
Great, thank you. So an additional question is, what might be the next steps to understand the effects of patient genetic ancestry, for example, African versus European, on the prognostic impact of somatic alterations in TLL?
It's another great question. I think one, we have to understand the biology of that. So why? So why, if you had a similar genetic alteration and your genetic ancestry is African versus European, what's driving that? We've started to look at this a little bit, and we don't need to understand why it is. So if you look at expression of Notch, signaling, downstream signaling, activation of pathway, what we found is patients with Notch, who are of African descent, who don't have a Notch mutation, have very low expression. If they have a Notch mutation, they have higher expression. If you compare those to European descent patients, those without a Notch mutation actually have that are European descent have similar expression as patients of African descent with a mutation. And then European descent with a mutation have a very high expression. So actually, the whole expression of the Notch signaling pathway is downregulated in patients of African descent. We have nice data and ex vivo data showing that Notch expression correlates with sensitivity to chemotherapy. So then the question is, what is turning down Notch signaling as a whole in patients of African descent? And if I was to be a betting man, I bet it's epigenetic, and it's one of the reasons we're doing methylation profiling right now, that it could be that we have learned that there is differences in epigenetic patterns of inheritances across ancestry. And one of our leading hypotheses is maybe patients of African descent are more likely to have hypermethylation of their Notch1 promoter that prevents transcription. And that doesn't matter what your mutation is if methylation is in epigenetic things preventing it from being expressed. That's just one. It could be that there's other pathways that are competing with it or feeding into it that are causing dysregulation. But this right now is an observation that we need to understand the biology of it.
Great, thank you. So an additional question is revolving venetoclax treatment. So you mentioned it was not successful in TALL in adults. Is that solely because of the adults do not have the specific BMP-like subset that has higher expression of BCL2, or are there potential other factors that you think are contributing?
So two things. One, it did have, so venetoclax was effective in some patients of TLO, both as kids and adults. So if I said that, then I misspoke. It's that venetoclax targeting BCL2 is really only effective in early lineage TLL and not as effective in later lineage TLL. And we've seen a little of that in our clinical trials as well. However, there have been some nice early phase data, mostly out of China, combining venetoclax that actually showed us pretty good salvage regimen. And I think it's just that, as far as what they're addicted to, is which patients respond to ven and which ones don't respond to ven. I think another nice thing is the patients who respond to dasatinib and have that elevated LCK and SRC signaling tend to be more mature TLL, and they tend to be mutually exclusive. So TLL that responds to ven tends not to respond to dasatinib and vice versa. So we're trying to figure out studies that we could actually figure out which ones are more likely to respond to ven versus dasatinib, and then actually get most of your TLL with one of the two targeted pathways.
Great. So we have time for about one more question. So the question is, was only NR3C1 expression changed in BMP-like cells regarding prednisolone or steroid sensitivity?
It's a good question. That was the main thing we started looking at. There's probably other things as well, but we haven't dove into them and looked at them. One of the things is there's a lot of questions to ask with a lot of this data. I think actually it's important that these data become publicly available pretty quickly. So the single cell data are on the Alex's Lemonade Stands single cell atlas site. Some of the bulk profiling data are in dbGaP. And there are a lot of questions to ask, and anyone out there interested in TLL should download the data and can ask some of those other questions. So if you're interested in steroid resistance, go for it.